Friends, welcome to this last video of this OER related to pharmacology of cardiotonics. In this particular video, we are going to see how cardiotonics exert their action. In earlier videos, we have seen that for maintaining the action potential across the cardiac cell membranes, different types of ion channels are functional. Now, in case of cardiotonics, these drugs particularly act by inhibiting the sodium-potassium ATPase channel. This particular figure is showing sodium-potassium ATPase channel along with sodium and calcium exchange. What happens in this case is these two exchange mechanisms are maintaining concentration of sodium and concentration of potassium across the cell membrane. In resting condition, amount of sodium outside of the cell is higher, the amount of potassium inside the cell is higher. Sodium amount outside of the cell is higher, potassium amount inside of the cell is higher. Now, if suppose there is diffusion dependent upon the concentration gradient, then potassium will naturally flow out of the cell and sodium will flow naturally into the cell because of concentration gradient. But this does not happen. These two pumps, these two exchange mechanisms maintain the concentration of sodium and potassium across the cell membrane in a particular manner. Now, if suppose we add cardiotonic drugs, digitalis-like drugs, which block this exchange mechanism. Which exchange? Sodium-potassium exchange, which is responsible for entry of potassium into cells, exit of sodium outside of the cells. If this process is blocked, this ATP-dependent exchange of sodium ions and potassium ions is blocked. Entry of potassium and exit of sodium ions, ATP-dependent exchange is blocked by these drugs. What will happen? Exit of sodium will be stopped here and entry of potassium also will be stopped here. Overall effect of this is the amount of sodium outside of the cell is reduced. What does that mean? Amount of sodium outside of the cell is getting reduced. Why? Because digitalis has blocked this process. Digitalis has stopped the activity of sodium-potassium ATPase enzyme. Now, if this happens, the amount of sodium outside of the cell is reduced, this exchange mechanism is blocked. Why? Because there is no sodium available to get exchanged for calcium. Now, whatever calcium was there inside the cell, it is remaining in the cell now. Okay? Whatever calcium was going to get exchanged for sodium, it is remaining in the cell because extracellular sodium ion concentration has got reduced. Why it has got reduced? Because this enzyme has been blocked by digitalis. Now, such accumulation of calcium because of inhibition of sodium-potassium ATPase enzyme will lead to release of more amount of calcium from intracellular storages called as endoplasmic reticulum. This is a folded structure, folded cell organelle present in the cell, which is calcium store. Okay. So, accumulation of calcium inside the cell because of inhibition of sodium-potassium ATPase triggers the release of more amount of calcium into cytoplasm. And such release now increases total amount of calcium in the cytoplasm. Okay. As calcium is responsible for muscular contraction, this accumulation as well as release from endoplasmic reticulum of calcium sufficiently increases the calcium ion concentration in cardiac muscles to trigger forceful contraction of the cardiac muscles. Now, this is how the failing heart will regain its ability to pump out the blood. If once this process is initiated, once the process of muscle contraction is initiated, this will mobilize the accumulated fluid in the circulation. So edema will be reduced and congestion in the circulatory system will be released. Congestion will be mobilized. And this mobilization of congestion will be treating the symptoms of congestive cardiac failure. This is how, by inhibition of sodium potassium ATPase enzyme, reducing extracellular sodium ion concentrations and accumulating calcium inside the cell, cardiotonic drugs improve contraction of cardiac muscles. The same has been told here. Cardiotonics inhibit sodium potassium ATPase enzyme. This enzyme is involved in passage of potassium inside the cell wall, sodium outside of the cell wall. If this process is blocked, extracellular sodium ion concentration gets reduced, and this consequently inhibits sodium calcium exchange. And due to this reduced concentration gradient of sodium, there is rise in intracellular calcium. This intracellular calcium releases more amount of calcium from inside store called as endoplasmic reticulum, and sufficient amount of calcium is available for muscle contraction. This is how cardiotonics exert their action. Apart from this action on sodium potassium ATPase enzyme, cardiac glycosides, particularly digitalis derivatives, they have other actions on the heart, like they reduce AV conduction. Means they slow the rate of conduction of impulses across auricular-ventricular wall or auricular-ventricular septum. Conduction of impulse from SA node to AV node to ventricles is reduced in presence of cardiac glycosides. These drugs also reduce resting membrane potential, means they increase the excitability of the cardiac muscle cells. Then they also activate latent pacemakers. Now pacemaker means that tissue which is having activity like SA node. These are the tissues which can initiate spontaneous impulses in the heart. Okay? So such spontaneous impulses may be generated in presence of higher concentrations of digitalis glycosides. And these drugs also increase refractory period of heart. Just in earlier videos we have seen refractory period is that period in which heart does not respond to external stimuli. Okay? Like this, cardiotonic drugs exert positive inotropic action and negative chronotropic action. Now these two words are important here. Inotropic action means ability to increase the force of contraction. So positive inotropic means increase in force of contraction. Negative chronotropy means decrease in rate of heart, decrease in heart rate. Okay, so these are actions of cardiotonics on heart. They have positive inotropic action and negative chronotropic action. This negative chronotropic action is because of vagal stimulation of heart or release of acetylcholine in response to presence of cardiotonics. Okay, so overall action is also, apart from sodium potassium ATPase enzyme activation, there are multiple actions like inhibition of AV conduction, there is activation of pacemakers, there is vagal activation leading to negative chronotropy. At the same time, there is positive inotropic action, means increase in the force of contraction. Apart from this, cardiotonics also activate renin, angiotensin, aldosterone system and sympathetic nervous system, and thereby they cause vasoconstriction. Cardiotonics also have mild vasoconstrictive activity because of this stimulation of sympathetic nervous system and RAS. Apart from this, they induce diuresis, means output of urine, output of sodium ions from the body, and they reduce the accumulation of fluid in the body. One important action of cardiotonics is also activation of CTZ, chemoreceptor trigger zone, in the brain, and because of this chemoreceptor trigger zone activation, the person consuming digitalis glycoside may suffer from vomiting and nausea. Okay? So this is how cardiotonics exert their action on failing heart, and apart from that, they have multiple actions on our whole body system. These cardiotonics, as they are increasing the force of contraction of heart and as they are reducing the body fluid volume, they are used in treatment of congestive cardiac failure. Friends, In class activity, we can discuss other aspects of cardiotonic drugs and their therapeutic uses. Thanks a lot for your patient hearing and visit to all these 5 videos. Thank you.